Okay, good afternoon students. In previous class regarding the cell, the unit of life, we were discussing regarding an overview of cell. In overview of cell, what else we are discussing? We are discussing the particular cell. Those are critical for the study of overview of study of cell. In the sense, one we have mentioned regarding the ODMP, another one is a human chip cells. Okay, and then we have studied about the cell. Usually, cell has cell wall. In plant cell wall is absent. In the sense, only cell membrane is present in animal cell. Then afterwards, we have studied about the cell consists of the membrane-bound organelle, dense membrane-bound organelle called as a nucleus, and based upon the presence of membrane-bound nucleus and non-membrane-bound nucleus, there are the two types of cells or two types of organisms. We discussed yesterday. ಅದನ್ನ ನಾವು ಡಿಸ್ಕಸ್ ಮಾಡಿದ್ವಿ. ಏನಂತ ಡಿಸ್ಕಸ್ ಮಾಡಿದ್ವಪ್ಪ ಅಂತಂದಾಗ, if the true nucleus ಅಂತಂದಾಗ, if the nucleus is membrane bound nucleus is present, that time we are going to call such type of organisms are called as eukaryotes. And if the true nucleus is absent, in the sense nucleus is non-membrane bound, okay, that time we are going to call them as a prokaryotic cells. Okay, then we have studied regarding the semi-fluid, in the sense we have discussed about fluid matrix filling in the cells that is called as a cytoplasm, and in cytoplasm we have studied the different kinds of cell organelles, and cell organelles again differentiated into two types. One is membrane bound organelles and non-membrane bound organelles, and I mentioned membrane bound organelles are present in eukaryotes, and in prokaryotes membrane bound organelles are absent. And non-membrane bound organelle, especially ribosomes, which are present in both eukaryotes and prokaryotes. And non-membrane bound centrioles, the cell division, those are present. Okay, and those are the in cell division now. So likewise, we are studying regarding the overview of cell in a previous class. Today we are going to discuss the basic things regarding a type of cell, means a type of organisms. Okay, we are going to study the type of cell, prokaryotic cells. How we are going to means prokaryotic cells, which organisms we are going to get the such kind of cells, and what are the basic characters of these prokaryotic cells. We are going to study in a present class. So usually the prokaryotic cells are represented by the bacteria. These cells are represented by bacteria, blue-green algae, mycoplasm, and PPLO. This PPLO is very important. One last question here. What is the long form of PPLO? PPLO is pleuropneumonia-like organisms. PPLO is nothing but pleuropneumonia-like organisms. These are bacteria, blue-green algae, mycoplasm, PPLO are going to represent the prokaryotic cells. Or a prokaryotic cells are represented by the bacteria, blue-green algae, mycoplasm, PPLO, etc. Now next, these are generally what? The prokaryotic cells are generally smaller and are multiplied more rapidly. What I told you, these prokaryotic cells are generally smaller and they are going to multiply more rapidly than, I written here, in front of this we are going to write eukaryotic cells. Eukaryotic cells, in the sense prokaryotic cells are generally smaller than eukaryotic cells and they are going to multiply more rapidly as compared to the eukaryotic cell. More rapidly in the sense multiply 1 to 2 to 2, 4 to 8, 8 to 60, 60 to 64, likewise they are going to multiply more rapidly. Okay, then you are going to compare with the eukaryotic cells. This is about what? The second basic character regarding the prokaryotic cells. Then next, there are different types of prokaryotic cells, or the prokaryotic cells are various. Okay, from means based upon their shape and size. They are going to vary based upon their shape and size. Now here there are 4 basic shapes we are going to study regarding the prokaryotic cells. What are those in the sense? First shape, it is a bacillus type of prokaryotic cells. that is rod-like structure. Coccus, spherical. Vibrio, comma-shaped. Spirillum, spiral. Spirillum, spiral. These are the four kinds of bacteria. We have four kinds of prokaryotic cells which are varies from one another based upon their shape. One is rod-like, spherical, comma-shaped, spiral. We are going to call it as rod-like in the sense bacillus, coccus, spherical, vibrio, comma-shaped, and spirillum, spiral. These four you are going to remember. Sometimes 2 marks question is going to ask what are the different shapes, write the name of different shapes of a prokaryotic cells. Okay, those are bacillus, rod-like, coccus, vibrio, spirillum. Understood? Next, what is the character of the next prokaryotic cell character is they have cell wall. All these prokaryotic cells are having the cell wall surrounding the cell membrane except myoplasm. In myoplasm, here I have the myoplasm. Myoplasm doesn't have any cell membrane. Doesn't have any cell wall. Doesn't have any cell wall in the sense. Apart from myoplasm ಹೊರತು ಪಡಿಸಿ ಎಲ್ಲ ಏನು 8 part eukaryotic cells are those are having the cell wall that is surrounded by the cell membrane. This is very important. Except the myoplasm. In myoplasm, cell wall is absent. Based on this, is asked the competitive exam question also. Okay, you ಬಹಳ ಸಣ್ಣ ವಿಷಯ ಇತ್ತು ಅದಕ್ಕೆ ನೆನಪು ಮಾಡ್ಕೊಳ್ಳಬೇಕು. Likewise, only they is asked the question. In the sense, which one of the following it doesn't have cell wall? myoplasm example ಕೊಡಬಹುದು, ಬಾಕಿ ಬೇರೆದು cell wall ಇದ್ದಂತ example ಕೊಟ್ಟು ಅದನ್ನ ನಿಮಗೆ ಗುರುತಿಸಕ್ಕೆ. That is like that is to remember. In the sense is to study point ಎಲ್ಲ ಪಾಯಿಂಟ್ ನಿಮಗೆ ನೆನಪು ಅದನ್ನ ನೆನಪಿಡಬೇಕು. Okay, then only it is possible to what crack the lid biology. ಗೊತ್ತಾಯ್ತಾ? So next point ಏನಕ್ಕೆ ಅಂದ್ರೆ cell wall ಬಗ್ಗೆ ಮಾತಾಡೋಣ. yesterday ಅಂತ ನಾವು ಎಲ್ಲ ಹೇಳಿದ್ವಿ cell overview of cell diagram. Overview of cell diagram ಅಂತ ಹೇಳಿದ್ರು fluid matrix filling the cell. Each both the cells eukaryotic ಇರಬಹುದು prokaryotic ಇರಬಹುದು they are going to filled with the fluid matrix and that fluid matrix we are going to call it as a cytoplasm. That we are going to call as a cytoplasm. And in the cytoplasm, yesterday I told you, cytoplasm consists of what? Dense nucleus, membrane-bound nucleus in eukaryotes. But in prokaryotes, there is no well-defined nucleus. There is no well-defined nucleus. There is no well-defined nucleus in the sense what? The nucleus is not surrounded by the membrane. Not surrounded by the membrane. So that what is going to happen in the sense? We are going to get the genetic material is basically naked here. Genetical material is what? Basically naked. In the sense, for example, suppose it is a cell of eukaryote ಅದಕ್ಕೆ ಏನಪ್ಪ ಅಂದಾಗ likewise cell membrane-bound nucleus ಇರುತ್ತೆ like this. But in eukaryote, this is eukaryote. Prokaryote there is no membrane-bound nucleus in the sense like this. You are going to get the nucleus in the cytoplasm. It doesn't surrounded by any membrane, so they are going to call this one as a naked genetic material. This to call that as a naked genetic material. Why it is naked genetic material? Because we are not going to found membrane-bound nucleus over here. In prokaryotes, there is no membrane-bound nucleus. That's what the genetic material is basically naked. And this genetic material is DNA. In addition to the genomic DNA, whatever the DNA is there in a genome or genetic material is DNA, in addition to that genomic DNA, you are going to have in prokaryotes single chromosome or circular DNA. Single chromosome or circular DNA. You are going to get single chromosome or a circular DNA. I told you here there is no well-defined nucleus. In the sense, nucleus is not covered by the membrane, and the genetic material is basically naked, and the genetic material is DNA. Addition to genomic DNA. Addition to the genomic DNA. What else you are going to find in a prokaryotic cell? One is single chromosome or circular DNA. Circular DNA. One thing we are going to study in these prokaryotic cells, that is what? Plasmid. PLASMIDs. This is very important, and this thing we are going to study next year. Biotechnology now this is a part of what? Genetic engineering. We are going to use this as a tool of a genetic engineering. Genetic engineering now next year study ಮಾಡ್ತೀವಿ. ಅಲ್ಲಿ genetic engineering ಒಂದು tool ಅನ್ನು ನಾವು plasmid ನ use ಮಾಡ್ತೀವಿ. Now here we are going to study about the plasmid. What is the plasmid in the sense? Plasmids are nothing but small circular DNA. Small circular DNA which are present outside circular DNA outside the genome. Outside the genome. Okay, or we are going to call this as extra chromosomal DNA. Small circular extra chromosomal DNA. Small circular extra chromosomal DNA which are present in the prokaryotic cells are called as plasmids. Plasmids are nothing but small extra chromosomal DNA. And these small extra chromosomal DNA are having a significant character like resistance to the antibiotics. They are having the resistance to the antibiotics, and in such a way that we are going to use these plasmids as a vector. Plasmids as a vector in the sense of transforming the alien DNA to the host DNA. We are going to study these things in detail in the next year in a chapter biotechnology principles and processes. So here we are going to remember the plasmid. Plasmid is nothing but small circular extra chromosomal DNA. Small circular extra chromosomal DNA which are present in a prokaryotic cells. Those we are going to call as a plasmids. Those we are going to call as a plasmids. Now another one important specialized character or a specialized differentiated form of cell membrane called as a mesosomes. Mesosomes is the characteristics of prokaryote. Mesosomes or mesosome is a characteristic of prokaryote. This is you are going to remember. The next one is they are essentially unfolding of a cell membrane. They are essentially unfolding of a cell membrane. Okay, this is about what the prokaryotic cells. In the sense, in prokaryotic cells, these many things you are going to remember. First, what is the important thing is that is pronucleus. Pronucleus in the sense nucleus is surrounded by the cell membrane. Another one is false nucleus in the sense the nucleus is not surrounded by any cell membrane. In the sense there we are going to find naked genetic material. Naked genetic material in the sense that is distributed in the cytoplasm. That doesn't have any boundaries. That doesn't have any boundaries. This is a very important thing. And here you are going to remember the long form of TPLO. TPLO in the sense pneumonia-like organism. This is asked many times for the 1 marks question. Then next we are going to study means the important thing is the different kinds of prokaryotic cells based upon their shape. Based upon their shape, there are the 4 basic shapes of the prokaryotic cell. One is bacillus, rod-like structure. Then punctus, spherical, vibrio, comma shape. Then spirillum, that is the spiral shape. Okay, these are the 4 what we are going to call it as a shapes. 4 basic shapes of a prokaryotic cells. Next, I told you all these prokaryotic cells are having the cell wall surrounded by the cell membrane except mycoplasm. In the sense, mycoplasm ಹೊರತುಪಡಿಸಿ ಬೇರೆ ಎಲ್ಲ prokaryotic cells ಅದರಲ್ಲಿ cell wall ಇರುತ್ತದೆ, and this cell wall is surrounded by the cell membrane. Next, inside that we are going to get the cytoplasm. I told you no well defined nucleus over there. That's what the genetic material is naked, and the genetic material is DNA. Addition to the genomic DNA, we are going to find the important that a single chromosome or a circular DNA in a prokaryotic cells. Then afterwards, the plasmid, that is very important. Plasmid is nothing but a small circular extra chromosomal DNA, and that has a particular characteristic. antibiotics, resistance to the antibiotics. Next we are studying regarding the specialized differentiated form of a cell membrane called mesosome. Is the characteristic of prokaryote, and then they are essentially enfolding of a cell membrane. This is regarding what the prokaryotic cell, the basic characteristics of prokaryotic cells. Okay, so in previous class we have discussed regarding the an overview of cell. In that we have discussed about the plant cell, animal cell, presence of cell wall, absence of cell wall. Then we have discussed about the prokaryotic cell, eukaryotic cell. In prokaryotic cell there is no definite nucleus, in the sense nucleus is not surrounded by cell membrane. And in prokaryotes, in prokaryotes we used to have the false, in the sense there is a true nucleus is absent, in the sense nucleus is not surrounded by any membrane. It is not surrounded by any membrane, and based on that we are going to discriminate eukaryotic cell and prokaryotic cell. Based on that we are going to discriminate eukaryotic cell and prokaryotic cell. Then afterwards, here in detail we are discussing about the prokaryotic cell. And I told you we used to write down few characters or few differences. You try to write down the differences between the eukaryotic cell and prokaryotic cell, plant cell and animal cell. You go through the diagrams which were provided in your NCERT textbook regarding the prokaryotic cell, and there they are mentioned some measurement of these prokaryotic cells. Okay, with this I am going to stop here regarding this today's class. In this particular class we will discuss about the prokaryotic cell and basic characters of a prokaryotic cell. Next class we are going to discuss the next point. So I am going to stop here with this prokaryotic cell. Thank you.